Hi everyone, welcome back. Carrying on with some organisation today, we're going to be looking at one of the core practicals, which is food tests. Now, food tests is a pretty simple practical to do. It just requires the knowledge of quite a lot of chemicals and reagents and things. So the practicals we're going to look at today is the Iodine test for starch, the Benedict test for reducing sugars, or Benedict's test for sugars, the Burette test for protein, and the Emulsion test for lipids. So, as always, grab some paper, grab those pens, and follow along with me. First test we're going to look at is the iodine test for starch. You might have come across iodine before. You will have used it in the first practical that you did when you made onion slides. It's that yellowy, orangey solution that turns black or blue-black when it touches starch. What we're going to be doing here is basically applying that knowledge to the practical. If you set out your ideas like me, you'll find that you make yourself a little results table using the IV and the DV arrows, because your independent variable is the thing you change. So that's, in our case, the food sampled, or it could be those categories. Dependent variable is what we're measuring to see that change. So that would be a colour change, whether something is present or not. The iodine test for starch is the simplest of the food tests, I find, because pupils have often come across iodine before. You might know iodine from the first practical, where you had to use it to stain some onion skin so you could see the nucleuses, or you might have even used iodine when looking at photosynthesis, when you might have dyed a leaf with iodine to see if there's any starch produced in it, and maybe compared that between plants that are in the sun and plants that are in the shade. The iodine test for starch isn't really very complicated. So for this experiment, you're going to need iodine, pipettes, food samples, and your spotting tile. If you can't remember what a spotting tile is, it's one of those trays that has all the little grooves, the little dots in them. What you need to do then is set up your spotting tile and put food samples into the trays. Then you're going to add iodine. Remember, iodine is orange, and then any foods it comes into contact with that has starch, that iodine will really quickly change into a dark black bluey colour. Any you see a colour change for, you just put that in your table. So if we were looking at potatoes we know contain starch, and we'd expect it to change to black, and sugar doesn't contain any starch, so there wouldn't be no change, it would stay orange. Okay, that's the iodine test done, so let's have a look at another. The next test we're going to look at is the Benedict's test for sugars. Sometimes it's called the Benedict's test for reducing sugars, but just remember Benedict's and sugar and you will be fine. Just as before, we need to look at our variables. So the thing that we're changing, as before, is the type of food we're sampling, and our dependent variable, surprise, surprise, is a colour change again. Okay, let's have a look at some different foods this time. Because we're looking at sugars, let's do a perfect sample, should we say, with foods that we know to contain different levels of sugar. So we're going to have apple, it's pretty sugary, it's a nice sweet fruit, potato, and strawberry, which are really, really sugary. And we'll have a look and see what we'd expect if we use these foods. The Benedict's test for sugar, I would say, is the most complicated to carry out. So what you're going to need this time is a selection of boiling tubes. You're going to need a water bath. If you can't remember what a water bath is, it's just the simplest way of doing it is having a kettle, pour some kettle water into a bigger beaker and float your test tubes in it. You're basically wanting to transfer the heat from the hot water to your sample, but you do not want to water it down. Kind of like how you would melt chocolate over a pan of water. Okay, then you need your food samples. So again, we're going to be looking at apple, potato and strawberry. Those would need to be crushed up and filtered preferably, but if you're doing it in school, it doesn't matter that much. Then you need some filtered or distilled water. It's important it's been filtered. That way we know that it is not something that's been added to the water that's caused the change. It's the fruit itself. And you'll need a little spatula for getting your food samples into your boiling tube. What you need to do then is, for all your food samples, have a boiling tube with your food samples in them, and then a little bit of water in top. About 20 millilitres will do. Then you need to make sure that those two things are mixed really well. You then add the Benedict's reagent. So this is why it's called the Benedict's test. Benedict's reagent is this lovely light blue colour. Then you put 2 or 3 drops into each boiling tube. You're going to put that into your water bath. It needs to be hot. The hotter the better, so about 95 degrees C. So it's really important, if you're doing it in school, you're always checking the temperature of your water bath water, and if it starts to cool, you add a bit more hot in. If you're lucky and your technicians are smiling upon you, you might be given a water bath that can be set and kept at 95 degrees. You leave it in there for about 5 minutes. when you are absolutely sure that the temperature of the food and Benedict's is the same as the temperature of the water on the outside. What you end up with is a set of colour changes that tell you how much sugar roughly is in each type of food. So you would have red for very high sugar, orange or yellows for middling sugar, greens for low sugar, and if it didn't change at all, if there was no sugar, it would stay the Benedict's blue. So if we go back to our results then, if we were to theoretically have done this with our 3 food samples, we'd find that orange, because it is high in sugar, would have turned the Benedict's an orange colour. Potatoes don't contain no sugar at all, but they're very low in sugar. Potatoes are a storage in plants, so they would have turned any sugars into starch to be stored, but there are still some sugars hanging around, so that's why it would have gone a nice green colour, so low sugar. Then you've got strawberries, who are famous for being really deliciously sweet. They would go a bright red, showing that there's a very high amount of sugar in there. Okay, that's the Benedict's test done. On to the Burette test for protein. Independent variable are the same as before. So the independent variable is the food we're sampling, and the dependent variable, we're looking for a colour change again. The method for this one is slightly less complicated than the Benedict's, but it doesn't involve any water baths. So you're going to need your Biuret reagent, so that's the chemical that you're adding and the name of the test. You need your boiling tubes, you need food samples, a spatula and some distilled water. What you're going to do first is put your food sample and your water into your boiling tube, same as we did with the Benedict's. Give that a good mix. Now, when you do the Burette test at school, you might be given your Burette reagent in 2 pots or 1 pot, because Burette is just a mixture of various chemicals to make it turn purple in the presence of protein. So whatever you're given, you need drops from 1 pot or a couple of drops from each pot into your boiling tube with your food. Then you're going to need to stir that for a couple of minutes to make sure it mixes well. I'd maybe use the spatula or a glass stirring rod. And then, if protein is present, it will change to a purple or a very light pinky lilac colour. And if there's no protein, it's going to stay the light blue that you started with. So if our food sample were egg powder, we would expect, because egg is a good source of protein, especially for vegetarian, that would turn purple in the presence of Burette's. Flour, on the other hand, has no protein, it's all just carbohydrates, so you'd expect no change. But if you see pink rather than purple, it doesn't mean the test's gone wrong, it just means there's protein present. Right, on to our last of the food tests, the emulsion test for lipids. Lipids is, of course, the posh scientific term for fat. And as always, we're going to look at our variables. Independent variable is going to be our food sampled, surprise, surprise. Our dependent variable is going to be colour change. So let's have a look at some foods. Let's pick butter, milk and coffee. Butter and milk, you'd expect to have some fat in. Coffee, if it's black coffee, I'd expect to have no fat in. So what you're going to need for this one is some ethanol, which is alcohol, some boiling tubes, spatula, some food samples and your distilled water. So as before, we're going to put our food samples and the water into our boiling tubes and make sure they're mixed well. Then we're going to add some ethanol. Now ethanol doesn't have a colour, it's colourless, clear. Mix it well, give it a good shake, but make sure you've got a bung on that boiling tube. If there's any lipids present, you should see either a layer or the whole solution will turn a cloudy colour, a whitish, yellowish. Kind of reminds me of the colour of mayonnaise, because when you make mayonnaise, you are making an emulsion. Butter and milk would contain some fat, so the butter would go cloudy, milk would go cloudy, though it might be hard to tell due to the milk's colour, that's why filtering would be important earlier on, and then coffee would not go cloudy at all because it's got no fat in it. Right, that's the food tests. So make sure you know for each one the independent variable, the dependent variable, some methods for each, what equipment you need roughly, and how you control those control variables. So things like keeping the same volumes of Benedict's in each boiling tube, making sure the same volumes of water, the same amounts of food in grams, for example. I find when I'm learning a method, if I make myself a drawn method rather than a written bullet points, it helps me remember. But if you want to turn my diagrams into bullet points because that works better for you, that is absolutely fine, whatever works best for your brain. Right, I will leave you, as I left you last time, with one of my bad jokes. It's going to be food related this time, so what do you call the cheese that makes you sad? Blue cheese.